right next to Acte.
I don't know any of these buildings. I just
How long have you been here?
I just started here, so like I've only been here for
makes sense.
few months at most. Okay. Anyway, so I tried to when I was trying to prepare, look up what you've been up to. I actually ended up looking at your lab's newsletter.
Nice.
It's a banger.
Well, I like it.
It's a banger of a newsletter. I actually didn't go anywhere else.
Okay. It is pretty complete. If you want to see everything we did,
it's incredibly complete. How long does this thing take to put together? Does everybody contribute to put it together? Okay.
Yeah, we ask everyone to write an update on their own projects.
Okay.
And we do collect pictures and stuff like that during the year.
Okay.
And then 2 people usually put it all together
okay.
at the end. Yeah.
Okay, so there's a whole bunch of stuff here that I want to ask. Anyway, I think your work is super cool. Because tangentially, I was sort of involved in the, in my bachelor's thesis. I did quite some work with dynamical systems, but that was mostly in video games and just sort of interacting people in the video games and how to study those things.
Mm-hmm.
And I got really extremely down that rabbit hole. And then for my master's, we also had an entire course on complex systems. So a lot of a lot of the metrics, a lot of the models and these kind of things. And then for my thesis, I did something on using EEG data to look at how using graph embeddings to see if they can improve the prediction performance on psychiatric conditions.
Mm-hmm.
So I was already very interested, and I was and I was just trying to just having a good conversation. So I just looked around and I found your stuff. But anyway, so a lot of stuff here. But
yeah, it's good that you found me.
Okay, so but let's start from the beginning, right? So how did you end up? Before saying exactly what you work on now, maybe just start from how you got there, because then that might make more sense.
Yeah, sure.
Yeah.
More chronological.
Yeah, exactly.
Sure. I've always been fascinated by human behavior, I guess, which made me want to study psychology.
Same. Exactly how I ended up here.
Really?
Yeah.
But we did take diverging paths.
Yes,
I do think because
paths, yeah.
I thought, I want to learn about behavior, and then I studied psychology, and I found out I do like anchoring psychology behavior to somewhere. So I did some explorations into social psychology and clinical psychology, but I really like psychology. So that's what I did. And then basically in the final year of my master, it was doctoral studies.
Wait, sorry to interrupt. Neuropsychology versus just psychology. What's the
neuropsychology.
Yeah, so what separates those two?
It's a part of psychology. So if you study psychology, it was at the VU. It was right here. Okay. If you study psychology, you get to pick one of 8, I think, back then, different directions. And it was the only university in the Netherlands that had a clinical neuropsychology as a direction, a program. Right. And so it was mostly about the relationship between behavior and the brain. So that's what I learned, and it's clinical neuropsychology. So I was trained to work in a hospital and do diagnostics and treatment for patients with brain diseases.
Okay. So how sort of the biology maps to directly to behavior in patients with some deficits or something like this?
Yeah, okay. And more or less the other way around. So basically you see a patient, you test their cognitive and psychological performances, and then you deduce what's wrong with their brain. Yeah. The field was a bit born before we had a lot of advanced imaging.
Oh, okay, yeah.
And to basically deal with brain diseases. So I think I studied in early 2000s. It was kind of the question, okay, what if you have someone with Alzheimer's or a traumatic brain injury or lots of developmental issues like autism or ADHD? What do we do with these people? Can we help them, basically? Can we find what's wrong or what they need help with and then treat them?
Mechanistically in the brain?
I mean, it's a clinical...
Okay.
It was a clinical track. So I was not on the pure mechanistic pathway in the coursework at least, but I think there was back then a pretty mechanistic localizationist
Right.
thinking about brain disease.
This part of the brain is responsible for this, and
Yes.
this causes this.
Yes, which didn't make a lot of sense to me.
Okay. Even
the
back then,
beginning.
you were like, you were feeling a bit like, what's going on here?
Yeah. Yeah, also because, I mean, I like the stories.
Yeah.
Like, we all know Phineas Gage or
yes, exactly.
HM with the memory loss,
Yes.
or, Broca and how he developed his
Yes.
theories about language. And I think to some extent they work. But then as soon as you see a patient, you lose trust. Or I lost the trust in that. So once you see patients, they're all different. They all have different issues. People with the same issue in their brain may have very different types of complaints.
Right. So people with like so just the people who didn't sort of get the whole Broca and the patient HM thing is like, these are the sort of cases that gave us examples of mapping a specific part of the brain to a specific deficit, right? Like memory or memory, language, recall, episodic memory, these kind of things.
And those cases will be presented as if that's like the mechanistic explanation. But once you go into the actual world of patients, the wide variety of patients with a brain-related disorder, you can see that these cases that confirm some sort of localizationism or mechanistic explanation are the exception.
Okay.
And the rule are people are just all over the place.
Yeah, so they have a deficit here, but then they have something completely
unexplained or whatever. And I think, I did my clinical internship first in the Slotervaartziekenhuis that's closed now, but an Amsterdam hospital.
Okay.
And I saw lots of patients with strokes, Alzheimer's, traumatic brain injury, and sometimes people had the same lesion location. We could see it on the scan, and still they would be super different. So that kind of sparked my interest into, what is happening in the brain? Why is this variation so large? And at the same time, I had been sort of interested in oncology for a long time.
Okay.
Just because, this idea of a brain tumor or like cancer in general, hijacking processes that are normally healthy.
Yeah.
Interested me, and also in clinical neuropsychology, most people would go either into aging populations, looking at neurodegeneration, or like kids for ADHD and autism.
Why those two like extremes?
Because they're most common.
Okay.
They occur most often. But I was more drawn towards acquired brain damage, and so oncology. So I did my final internship on brain tumors, and it was kind of lucky that I found Kees Stam, who was my promoter, who had been basically one of the founding people of this field of network neuroscience.
Okay, wow, okay, nice.
Yeah.
That's perfect.
So that's kind of how it all happened, because then once I met him and we talked about like how the brain could be a network instead of mapping one-to-one function to a lesion or a brain location.
Yeah.
Once I found that, I thought, this is how it works.
I mean, this sort of kind of makes sense even without any of the because we know, for example, that if you have, for example, if you're blind, you're born blind, there's no real estate that's wasted, right? Real estate gets co-opted to do other things. Yeah. So doesn't that sort of already give you a hint that maybe the location is not that important?
I think the truly mechanistic localizationists, they think that that's development.
What do you mean by development?
Like plasticity is, if you're born blind or you're born with something, then of course development impacts that.
Okay, yeah.
So I think they only start counting from the adult life and then looking at it from the perspective of mechanisms. Okay. But you're right. I think it doesn't help for the whole story. Yeah, and then during this master project, I correlated global network topology measured with MEG to cognitive complaints, and they were the correlations were much better.
Yeah. Okay. Nice.
Yeah, and then and then I thought, network science is it.
I mean,
kind of blew my mind for in general. Like I have no maths or physics in my background, so complexity was not something I had met before. And then I thought, oh, this is how the world works. It was kind of a breakthrough moment in my kind of
like,
thinking.
It's actually like, because I've only very like, okay, so there's like this entire like network sort of graph theory, network stuff. It's this entire field, right? It's like this crazy rabbit hole. And, I just like touched just a little bit for my master's thesis, just to know enough to know, okay, what are sort of like the pitfalls. So like, what should I normalize? What should I put here? How do I represent this? Just enough to know that, right? But I know there's an entire like whole field about this. Yeah. And I find it very interesting because in terms of like the different kinds of abstractions you can use to model the world, a graph and a network is so like it is seamlessly applicable to so many different things. Yeah, it's almost like for a lot of things naturally falls out that graph structure is actually the best thing to do to represent it, like friendship networks or like traffic networks or paper networks, anything like this. Right? But yeah, like, and given that it's the brain, and we have known the interconnected nature of neurons and the whole thing for a very long time, it's kind of like, okay, of course.
Yeah, made total sense to me.
Okay, nice. Yeah, like, so for your master's thesis, what exactly did you do?
The question was, can we better understand cognitive deficits in brain tumor patients by correlating them to the global brain network topology, so to say. So basically we did MEG in, I think it was like 20-something patients, all with a slow-growing brain tumor, and they all had some sort of cognitive deficit. So I did a extensive neuropsychological battery. I tested them in their homes. And
wait, how did you do this? How did you, I'm trying to realize sort of like how awesome it is to sort of be in sort of this little area of Netherlands, or like you can actually get patients for your master's thesis to actually like do MEGs. That's crazy.
Well, they came from all over the Netherlands, actually. Cool. I traveled, so that's the reason why they asked a student with, you get the free public transport pass. I don't know if that's still the case, but I think it's still the case. I had it back then, and then I had to travel all over the Netherlands. So from Rotterdam to Putten to...
MEG at home?
No, I did a neuropsychological assessment. They did come here, but actually brain tumor patients get scans every 3 to 6 months.
Okay, so I don't know, I mean, I'm just now getting into because I'm from a purely machine learning background, right? Yeah. Like, so all I knew was that from like the from the first year of my master, I was like, okay, I'm going to focus on healthcare and medicine and health. Like, let's just go. And I basically tuned my whole thing to that direction. And from what I understand about cancer, I don't know anything about cancer. Okay, I'm still learning. All I know is I read like one book about it. But
which book?
The Emperor of All Maladies.
Yeah.
That's a good one.
It's a good one.
That's such a good book. Yeah. Like I couldn't believe I read a 1,000-page book on cancer. I was just like, this is so good. This book. Anyway. Yeah. But brain tumors are very rare.
Yes. So, well, actually, there's lots of different types of brain tumors. So let me separate them. So the most frequently occurring type of brain tumor is a metastasis. So that there's a tumor or there's cancer somewhere else, and it spreads to the brain. Yeah. So lung cancer, for instance, breast, prostate, melanoma, they all spread to the brain.
This podcast is extremely selfish in the sense that I'm just following my own curiosity, right?
Sure.
Okay, so if I understand metastasis correctly, it's like the multiplication of the cells. Uncontrolled multiplication starts somewhere else, and then those quote unquote bad cells somehow enter the bloodstream or the lymph system or something, and get somewhere else. Yeah. And they start multiplying somewhere else, and then they start breaking that place down. Yeah. That's the general vibe, right?
Exactly. Okay. Yeah. So the cells that you see in the brain, if you take it out, if it's a metastasis, it will have characteristics of the original type of cancer. So you will find, if it's a melanoma, you'll find melanoma cells there. Okay. So that's what we call secondary brain tumor, because it doesn't originate in the brain, it's a metastasis. That's not what I study in general. So I'll leave those out for the remainder. Then there's primary brain tumors that are inside the skull, but are not from inside the brain.
So if I understand correctly, the reason like there are no like neuronal cancers is because neurons don't divide after?
There are neuronal cancers.
Oh, okay. Well, I mean, there are, okay. I have no
But
idea.
typically they occur in kids.
Kids, right? Because as you get older, the neurons aren't dividing as much or at all.
I think it's still debate whether
Okay.
neurons can be born at later
okay.
ages. But
I know plasticity is like there's a lot of work, but like it's still plastic. You can still learn things because I know for a while, at least it was like once you get past a certain age, like that's it. You can't learn things anymore. You can learn
that's
things,
ridiculous.
but like the plasticity sort of gets harder to do, and things are more difficult to learn. And sure, I think I sort of get by that, but also like as you grow up, you have other priorities, right? And you get different kinds of reinforcement. Like because I was at Tilburg University, and there's a lot of work on language learning and stuff going on there. And from what I learned there is just like, for example, if you're an adult, the amount of corrective reinforcement you're getting. So I'm learning Dutch right now. Right? No one's on the streets
That's
like,
hard.
hey, let me correct your Dutch. No, that's wrong. Say it this way.
Yeah.
Like when I'm a kid, I was like, everyone's trying to help you.
Yeah.
But
well, and if you're a kid and you're learning a language or multiple languages in like a crucial window, we do know that your brain is really attuned to language in that phase. So it is more difficult to learn a language in a later age. It doesn't completely go away, but the neurons, at least in the cortex, so that will be the sort of gray matter of the brain that is on the outside, they don't divide. So there's very few brain tumors that are occurring from neurons. And if they do, it's mostly kids. And I don't look at kids. So because it's a different type of disease. So I'll leave them out as well. Then there's the meningioma. So those are inside the skull, but they don't penetrate the brain tissue itself. Because
covering.
So the meninges is kind of the lining of the brain. So they do put pressure onto the brain. People can have complaints. I have studied those a little bit. Interestingly, and again, sort of negating this whole localizationist theory, these people also have lots of cognitive complaints, even if you take out the tumor. These are typically like tumors you can treat really well because they're on the outside usually, so you can operate, and then they're just gone. But patients have a lot of complaints.
Do people like respond well to that? I mean, how good? What is like the survival?
That's normal.
Normal, okay.
Like you and me, except if it's a very rare, like high-grade tumor, as we would call it. But most people with these meningiomas, they have normal life expectancy.
Okay.
Many people have them without knowing it, actually.
Right.
We estimate that maybe 10% of the normal population has one of those.
Wow.
Could be probably very small and not give any complaints.
Okay. Any sort of like, but okay, let's think. When you say complaints, could it be this like so imagine you just had it for so long.
Yeah.
This is your life now.
This is what fascinates me most about this whole brain tumor business. Because, and let me get to the tumors I do study, which are very rare, which are gliomas. So they grow from the glial cells of the brain. They're supportive tissue. So you have the neurons that are the main sort of cells doing the electrical activity in the brain. And then you have astrocytes and oligodendrocytes that are sort of supporting the neurons. So they're feeding them and putting the myelin. So that's kind of the insulating.
Are they also the ones that they're responsible for the myelin sheath, or like
that's the oligodendrocytes?
Yes.
Yeah, okay. Exactly. So they're supporting, and those are vulnerable to glioma. So that's brain cancer, basically.
They divide and multiply, so they
all the time. Yeah. Except actually, I'm learning more about this because I come from the psychology side and the imaging, but now in my team, we also do a lot of lab work. So I have an expert, two experts on oligodendrocytes.
Super surprised by like a newsletter. I'm like, oh my god, you guys are doing a ton of wet work.
We're going all over the place.
Actual with there's this one thing about somewhere you messed up some bleaching, and I was like,
yes. They're staining the oligodendrocytes, and actually, I've learned now in the past 2 years or something like that, that normal oligodendrocytes don't divide in the brain.
Okay.
Not typically. But they do change a lot. So it's the same cells, but they wrap around one axon. And then if you learn something, for instance, the idea is that certain axons are kind of strengthened by learning or by adaptation. So then the oligodendrocyte starts myelinating some different cells.
Yeah. So when we say learning, I think maybe people sometimes think like when you learn something, there's some new neuron that is created to like learn. It's more like the connections between the neurons.
Exactly.
Updated, right? Yeah.
And probably not really the connections, because axons, which are the kind of linking pieces of the neurons, they don't go to completely new places, especially the longer distances. So they kind of are made when you're still in the womb.
Yeah.
Then there's a lot of them up until like 7 years of age, and then there's pruning, and then a lot of them go away, and you can have new or axons kind of refocus, I think, close by, but not at a distance. Okay. So it's not like, oh, you need a new connection between the motor cortex and the visual system.
Right.
You can't grow an axon there.
Right.
It's already there, and then sort of these supportive cells are probably going to strengthen or tone down the connectivity of these different cells. Okay. But this is all, I mean, I'm saying it as if this is something we can test.
Right.
We cannot, at least in humans, right? Okay. Because you cannot observe single cells in humans. Yeah. So you need, very, very rarely can do that. But you can't look at like the dynamics of axons. Yeah. So that's all either in vitro studies with cells or animals. Okay. Yeah. So these brain tumors, gliomas, they're rare, very rare. We have about 1,000 of them per year in the Netherlands, new cases.
So they're like extremely deadly, right?
Yes. So unfortunately, I will say that there's 4 grades, according to the World Health Organization. Grade 1 is kind of not growing, typically. So they are also benign, just like the meningiomas. So I will also leave those out. Okay. They are extremely rare. So if you hear things like centroastrocytoma or ganglioglioma, they are very specific subtypes of glioma that are grade 1 that don't really grow, and you can treat them relatively well. I mean, people have epilepsy most of the time, but the life expectancy is fine. Okay. So I will talk only about 2 to 4. Yeah. And those are unfortunately very difficult to treat. Okay. So grade 2 and 3, we think that life expectancy is about 5 to 15 years. Okay. Wow. On median, of course. That says nothing about the individual, but the median is like that. For glioblastoma, which is grade 4, which is the most frequently occurring, so 70% of all gliomas are glioblastomas, the life expectancy is way shorter. Okay. Yeah. Wow. So that's hard. Yeah.
So like, how do we get here? Yeah, your thesis. So your master's thesis. Okay. So, so there, did you look at a specific type of
low grade glioma? So that would be grade 2.
Okay. Yeah. And what was and you wanted to and what were the questions you were trying to answer?
Well, it was the second time that we applied network theory to brain tumor patients, but in general, this was 2006. Yeah. So the field of network neuroscience was kind of
I can imagine
being born. I would say 2005 was the first paper by Olaf Sporns who mentioned the term connectome, for instance. So it was really all new. So it was very descriptive. So the question was, does network topology, and I looked at clustering coefficient and path length, do they correlate with cognitive deficits?
So, like, maybe you want to say a couple of things about these measures, like, because there are all kinds of ways to quantify and like actually put a number on a on
now there are. So back then, if we go back in time, 1998, we had the Watson-Strogatz paper, who of course looked at how can we analyze networks. I think that was the birth of network neuroscience ultimately, and all sorts of applications of network science, because before that it had been used in social sciences, right? So we know the crucial experiment with the letters by Stanley Milgram. Have you heard about this? The 6 degrees of separation.
Yes.
He sent letters to people in the US and asked them to send it to someone they knew on a first name basis in order to get closer to this one person in Boston that it had to be sent back to. And then it turned out that most letters sort of hit the target within six steps. So 6
crazy.
Degrees of separation.
That's actually, actually, I knew about this idea, but I never knew about the actual experiment.
Oh, really?
Yeah. This is the
1967. Stanley Milgram.
So amazing. So he basically he wrote a letter to person and then told them to send it to another person.
Yeah.
And then his goal was to get the letter to one specific person.
And no one knew that person personally, but
Yeah.
they could only send the letter to someone they did know on a first name basis.
Okay.
Who they thought could be closer to the one person that was the target.
Oh wow, that's crazy.
And then it took 6 steps.
That's so
on
crazy.
average.
Yeah. Wow.
Yeah. And it was hundreds of letters, and
Yeah,
I think half of them made it over the years, because it took years, of course, back then to send letters.
That is such a cool experiment.
Yeah.
I love experiments like this, where it's like it's not like some crazy math, it's not like some crazy abstract 50, like there are obviously there's a place for that kind of experiment.
Mm-hmm.
But something like that that's so simple. It's like, you know what? Let's just send some letters to people.
Yeah. It's really awesome that he did this.
Yeah. That's so cool. Yeah, that's incredible.
Yeah, so that's the path length basically, right? So the number of steps it takes to get from one node, or one, in this case, person in a network to get to another would be the path length, which has been studied since in social networks a lot, I think.
Right, yeah.
But there's this other kind of, so it's a measure of integration, right? How integrated is this network?
Yes.
If there's very few steps connecting all people, we know that it's integrated, and it makes a lot of sense, especially now that it's much lower than 6 through the internet. So that has been studied a lot in the decades after as a separate measure, like how integrated is this network. But something else that people notice as soon as you look at networks in life, either biological or man-made, social, doesn't matter, you also have cliques. So you have groups of people or nodes or proteins even that are more connected to each other than to the rest of the network. Kind of like clusters or communities.
So if I have one of my friends is more likely to be connected to another one of my friends.
Exactly.
Right? So it's like a clique, like high school.
Yeah,
right?
exactly.
With friends, and then maybe one person in our little group is like super hypersocial, and he has connections to all other kinds of groups all over the place.
Yeah.
So he's like that one person that sort of takes you from this clique outside to another clique, maybe.
Yes.
Okay.
Exactly. Yeah. And ultimately, in a social network, everyone has cliques, right? Because you have a school clique, but you also have a family clique and a sports clique, and I know you're describing now something who's hypersocial.
Yeah.
That will be a connector hub, but people always have sort of connections to different clusters.
Right.
So that had been observed in real life networks, and I think measured to some extent. It's a measure of segregation or specialization. And I think up to 98, it was kind of unclear how these two interrelated. So they observed, path length is usually low, integration is usually high in networks that are working, and clustering is high, but how does it work? So then Watson Strogatz really defined these two network measures, path length and clustering coefficient, as a function of the connectivity of the whole network. And this topology where a network combines both high local clustering, so there is lots of cliques or communities, with low path length, so high integration, is called a small world network.
Right. Yes. So like basically you have, so is it kind of what I just described? It's like there are small clusters of really highly dense little population, and then there's like you can get to every other cluster in the network, but it's not the case that everyone in the network is connected to every other cluster directly.
It's kind of like the train network in the Netherlands, if it runs.
Yes.
You have these local trains, right, sprinters, that stop at every little station, but you also have intercities that connect the major cities. So typically, if you want to get from a small place to another small place, you first take one of these sprinters, then an intercity, and then a sprinter again. Right. Okay. And that is kind of the idea.
Okay. And then, so when you wanted to think of the different parts that are communicating in a network, how do you separate the parts of the brain out?
Yeah. So we use parcellations or atlases. So
like anatomically described areas, or?
We started with that. So back then, in 2006, I used an anatomically based atlas based on cytoarchitecture. So like in postmortem brains, they looked at what do the cells look like under the microscope and where does the look of the cells change. And then divided the brain into 78 regions. and that I used as nodes in the.
Is that still like
network.
the sort of?
No, now we typically use a mix of anatomically based, but also connectivity based.
Okay,
so people have looked at.
Oh, okay, so you were that's interesting because I didn't I just took the connectivity based idea for granted for because I just realized now that like you were there when there was no idea of connectivity to put.
We didn't have it.
Didn't have it, so you just used the location. Because I remember thinking for like one of the things I suggested in my thesis as future work is because I use connectivity as just distance. Like I used the electrode position on the head and then just looked at distance based.
Done that too.
Yes, but then you can do. Then I was like, okay, we can actually do connectivity based.
Yeah.
And I suggest all these other metrics, and now I'm realizing, there was a time where those didn't exist.
Yeah, the two interact. So you can, once you know connectivity between different regions, you can parcellate the brain differently. So typically what the atlas we use now would be based on, like, is the connectivity of each voxel, and each voxel represents like a little cube, like a pixel, but on a scan of the brain. Is each voxel unique in its connectivity profile? Because we do think, we can measure with really high resolution now, less than 1 millimeter squared, but we don't think that connectivity
many neurons
is...
are in one
the neurons?
In 1 millimeter cube?
Millions. Yeah. So it's very general.
Okay. Is it still like mostly cortex-based stuff, or do you have is there also work in more deeper sort of like the other
subcortical gray matter?
Stuff.
But
those ones
for sure. And with MRI, it's easier to measure those deeper structures. During my PhD and the master thesis, we used MEG. Yeah. Where it's harder to go to the deep gray matter, just because we're measuring outside the brain and we have to source localize the activity back into the brain, and that's harder to do for the deep gray matter.
Okay. So you just use you use 78 different regions of the brain
as nodes, and then as connectivity measure we use I think back then it was the synchronization likelihood.
Mm-hmm. How likely was one part to be active as the other? To the other parts of the brain.
Yeah, it's like global coherence. Yes. Kind of a correlation measure.
So you have a correlation. So if you have like a matrix, it's like you have a correlation between one part and every other part.
Yes, because these time series do have very high temporal resolution. Okay. So with MEG, but also EEG, you measure like 1,000 times per second. Right. So your sample frequency is really high, and you can discern different frequency bands in the brain from like half a hertz to... I think in my thesis we did up to like 60.
Right.
Yeah.
Okay. And then you just showed that look the pattern of activity. So you have this 78 different regions. Then you look at how synchronized the activity is between each of the regions, and then you show that.
Make a matrix.
Exactly, and then you have like one. You compress that knowledge, that information into a number, and show that. This model and the behavior and the correlation of the model is better fit than just like a purely location based sort of explanation of why this deficit occurs. And I know I'm butchering it, but like
I don't think we directly compared that, especially in the beginning, because it's just hard to say based on location of an individual tumor. What the deficit would be. But we basically extracted the clustering coefficient and the path length for this network, correlated those to the cognitive deficits, the extent of them, and then found a correlation. So lower integration is related to cognitive deficits and to some extent higher segregation. But the integration is more consistently correlated to the cognitive deficits.
Okay, nice. Yeah. So that was so after your thesis, so that's where you started, right? And then, so what next?
Yeah, I was in doubt a little bit in that phase of life about because I did clinical neuropsychology and I did like the hospital and seeing the patients, but so I was applying to get a job as a psychologist, which is now and was back then impossible.
So I was kind of, I was working in a theater, but I was kind of not doing what I wanted to do for some time. But I kept in touch with my supervisors for the thesis. And then at some point, they basically offered, I actually had a job interview somewhere else for a PhD position, and I was in the final round. Okay. And then I told the people here, my supervisors, and then they were like,
now or never.
Let us make some phone calls. And then the next day, I had an offer to stay for a year. So they couldn't pay for a PhD. But they scraped some money together and then said, we're going to pay you for a year to do research. But you will have to get your own funding in some way. So then I just started doing research. And there was lots of data for that back then. These were all like sample sizes of 10 or 15 patients. But back then, because it was also new, this worked out great. So I could just analyze.
So just because it's sort of related to like what I work on, what I'm doing, because in Mark's department, like in our department, it's like, so a lot of we work with a lot of problems with like very small sample sizes and thinking about how but very high dimensional data. You have like you said, like 10 patients sometimes. So how do you sort of even get like statistically like good
back results? Then? Now we have much more. I mean, it worked. So the thing with small sample size, of course, is that sometimes you overestimate an effect and you can't replicate it. But if you can, which we did, and the effect is just very big.
True.
Then you find it.
That's
so
true,
I think that was the case for both the integration part, because we keep seeing that even up to now. And it's not specific to brain tumor patients. So people in general, I also did a study in healthy participants back then, and also more recently, people with more integrated brain networks across different modalities are just smarter in general.
So integrated meaning like
path length, literally path length, or
so shorter path length. Wait, no,
is better.
Wait. I need to get my head around this. Okay. So
use your path length.
so wait. Let's see how short my path length is. No, but okay. Wait, I missed something here. So can we just define again what integration is? So integration is how tightly coupled the different areas of your brain are in activity, or?
Yeah, so you can define it in different way.
Okay.
And we started looking at the path length. So then shorter is better in general.
Okay.
In healthy people for sure.
Yeah.
But later on, we made it a little bit more sophisticated. So then we look at hub regions and how central they are. So we would, for instance, use between centrality or eigenvector centrality, because we know that these regions play a major role in kind of diffusing the information across the network.
Okay.
And so those are the major connector hubs that are crucial for information processing.
Okay.
So then we can also look at centrality of those regions.
So when you say somewhat higher integration, do you mean there are fewer long-range jumps that you have to do, or?
We don't actually take the length of anything into account. So it's purely based on the connectivity measure. So back then it was the synchronization of the time series of activity. But we've also seen this using diffusion MRI. So then you look at the sort of integrity of white matter connections. So then you look at large bundles of axons, and we see the same thing. So better myelinated or more sort of integral anatomical connections between brain regions. If you have those all over the brain and your path length is short, you're probably smarter.
So I'm still wondering, what do you mean by path length is short?
It's a number of steps. So if you think back about the Stanley Milgram experiment, it took 6 steps on average. But it would say that if, let's say your brain is this experiment. If you have 6 steps, your IQ would be 100. If you have 9 steps, it will be like 70.
So 9 steps meaning...
you have to take more steps on average to get from one side to the other.
Oh, okay. So from the signal to travel to one...
That's the hypothesis.
Exactly traveling here? That's the confusion. What exactly is traveling between these
Yeah,
parts?
this is a great question and one of the most fundamental discussions in the field. Okay, great. So
we'll answer that right now and then we'll sort that out.
Well, I don't know if I can quick answer it with a definitive answer because it's hard to study. But in graph theory, the path length would be a number of steps. On average, it takes from one node to the other. Yeah. In the brain, we have a problem because we can measure these white matter bundles, and some people think that they are physical connections and they're 100% true. Yeah. It may seem like that, especially to physicists, like, oh, we should take the white matter because that's at least a physical connection, which is what graph theory is based on. Yeah. I don't agree fully because it's a reconstruction. So you measure the diffusion of water molecules in the brain, and there's room for error in that. The signal-to-noise ratio is definitely not perfect, especially in some regions of the brain. So it is a reconstruction of these structural connections. Okay. But then what we are typically looking...
But when you can't just open a diseased brain and look at you can see where the white matter is and where the connections
You mean
are.
a dead brain?
Yeah.
People have done that. It's hard though, because the tissue disintegrates.
Really? That quickly?
To some extent, yeah. Wow. And you still, there are anatomists in our department who are very good at doing this with postmortem brains, but it's very hard to dissect individual cells and axons. Yeah. Okay. So it will be very hard to make a quantitative measure of how many connections are between different brain regions. Okay. And then, of course, a dead brain is a dead brain. So these people are typically, we have a collection of non-neurological donors. It's led by my colleague Laura Jonkman. So they ask people to become a donor if they don't have any neurological disease, and they typically die of something else, not related to the brain. Yeah. So they're healthy brains, but the person has died. Yeah. So it's not... so they're typically much older than the random person you want to explain the brain away. And there's lots of variation again. And this is why it correlates, I think, with intelligence to some extent as well. That there's lots of variation in the brain. Yeah. And intelligence is one of those things that correlates with how your brain is organized.
Right. Wow.
And so you can't look at one brain and then say this is how it works
this is
forever.
News to me. Yeah. This is like this the correlation between intelligence and the sort of the connectivity connectome. This is news to me.
Oh,
I had
it's pretty consistent.
So how do you
it doesn't explain everything, right? Okay. So it explains maybe 20 to 30% of variation in IQ, which is a lot because brain size does not.
That's a lot. That's like the brain size. Yeah, brain size does not. Yeah.
No, so it's considerable, but not everything for sure. Yeah.
Wow, but that's a lot. That's quite a lot. Like for IQ, there's so much research on IQ, right? Like people, try of AI aside, just like it's probably like one of the most researched areas in psychometrics, like quantifying IQ, figuring out how smart people are.
I don't really like IQ, to be honest, as a test, but there's lots of discussion on what it means because it has a very high propensity towards more information processing speed. Yes. So I don't like it from a clinical perspective, is what I should say.
What do you mean? Can you say more?
So patients typically are slower than average. And if you only use IQ type tests, they will perform worse always.
But can't you just take the... I see what you mean. So if you take a Raven's progressive matrices.
That's different, but that's more fluid intelligence, which is not super sensitive to brain disease. Yeah. But it's also difficult to say because, of course, educational level plays a huge role in all of these things, which is something you want to kind of factor out of clinical neuropsychology because you want to see what the impact of the disease is. Not
because I guess most IQ things are time-based, right?
Lots of them.
Yeah. So you have to do a certain thing in certain time. Yeah. And of course, I can see how that. But. So wouldn't a more integrated brain be faster?
It is.
Okay. So
that's saying the same thing basically. Yes, your IQ is higher, but you're also faster. And if you look at more clinically relevant cognitive tests, people also perform better, specifically in time tests, but also executive functioning, which is
just controlling your actions or decisions.
Yeah, it's kind of if there's a pyramid of how high level your cognition is, executive functioning would be on the top, because it asks that you not only process information that comes in, so input from your environment.
You do also learn what to ignore and what not to
also
use.
So a component of executive functioning would be to ignore certain things, like in the stop signal task, you have to ignore distracting input in order to pick the right output. But it can also be like working memory, so you have to hold in your sort of cache some numbers. Like I could read you now 7 numbers and you would have to repeat them to me or repeat them backwards, which is harder, obviously. But people can do that really well in the test setting. But that's an example of executive functioning. And another big component is cognitive flexibility. Right. So once you have something in your system, once you've learned something, taking an input, doing something to it, and then outputting it, changing the rules.
Okay.
So you might have heard of the Wisconsin card sorting test. So that's a test where you you show them cards and they have to sort them. And there are 3 different rules by which you can sort them. And you start with one rule. For instance, it has to be based on color. Yeah. So the signs on the cards, you have to put them in the right color bin. But then once people have learned that that's the rule, you change the rule. And then you go into a different...
And you measure how quickly they adapt to the new rule? Okay.
Yes, it's not a super sensitive task, I have to say. So typically you do measure how long it takes, but most people can do this, actually. Right. Yeah. But people with brain disease can be...
you can
lose disability.
I see. Yeah. So you can... So what are So is this one of the go-to techniques to sort of look at to sort of understand the deficits of somebody, or
we typically do a range of tests.
Okay.
Instead of just one. Okay. So we typically don't do Wisconsin card sorting because it just takes long and you can't really repeat it, because once people have done it once, so you can't measure change at all. We do the Stroop test, which you might have heard about. So that's cognitive flexibility again. You have to not read a word but say the color of the ink that it's written in.
That's super.
It's very hard.
It's very hard test.
So you have to inhibit the sort of reflex to read. Yeah. We do a version of the trail making test, which is set shifting. So you have to go.
Hearing about all this, I'm starting to get very worried very worried about myself. I'm thinking about I'm quite bad at some of these things.
Oh, but I think you wouldn't be, actually. Okay. Most people are pretty good at these things, especially if you're educated.
Yeah, the Stroop things, it's awful. I've done it because I've done it for little experiments and for these things.
Yeah.
Oh, it's so demoralizing. But also
it is. It's hard for everyone because you constantly have to inhibit your response. So it feels really not chill, but people do it. People with dementia obviously have lots of issues with this, but in general, people can perform pretty well. And a lot of brain tumor patients also perform really well on these types of tests. We do typically a whole battery of different tests to account for different types of cognition. Okay. Yeah.
Okay. And why? So why? And of course, you're in the ADORE group. So UMC, you're focusing on cancer. Do you still think that's pretty interesting? And there's a lot of work to be done there, or
for sure. Yeah. We are barely scratching the surface. Is my feeling.
Yeah.
So over the years, we've learned some of the more consistent things, if your brain is more integrated, it generally does better. Yeah. But we've also learned that that's not always the case. So people with epilepsy typically have higher integration, which is not helping.
Yeah, because I would imagine epilepsy is sort of like this traveling
hypersynchronization,
hyperactivity, right?
Yeah.
And then if you have super highly connected, you just get seizures all over the place.
Yeah, for sure. Okay. So that's not good. Yeah. It's also not good to have too high a clustering in general, is what we're seeing. And we're not sure whether that's because of the clustering being high, but because of the combination with low integration. So these things typically go hand in hand. So what you basically see is that a healthy network is kind of modular, and it has some hubs.
So basically, it means,
modular meaning some parts.
yeah,
do
modular
some
means
things,
that so you have clusters which are typically viewed as small groups of nodes in a network, like 3 or 5. But you also have modules, which is typically larger numbers of nodes.
Okay.
So, for instance, in the brain, we have these subsystems that we all know. So like the motor system would be a module. The visual system would be a module. We have the default mode network, which a lot of people think will have heard about, which is a distributed network that spans basically all lobes of the brain, but it's consistently connected to itself. So it's constantly communicating with itself, or at least connecting. And these modules are organized in a hierarchical way. So if you divide the default mode network further, you will find like 3 other smaller modules.
Okay,
so there's kind of a tree-like organization to the brain, where you have some regions that are just very connected to a lot of different things. Then you have a second sort of level of nodes that are also connected to many things. And these would probably be like the Rich Club, which is this definition made by Martijn van Heuvel, who's also here at the VU. Of nodes that connect primarily to other nodes that are highly connected.
Like when I think of when I think of like these sort of hierarchical structures that sort of like connect upwards or connect upwards, for example, let's say, like my first thinking is like a filter of some sort, like
Yeah, that's from the neural network, but these are all bidirectional. They go all in all directions.
Yeah.
And I'm presenting it as a tree now because it's easier to grasp, but it's messy. It's not like you have this one node on top and then it goes into 2 and then it goes into 4. It's a little bit wonky. But the idea is that there are several sort of layers. I don't necessarily want to call it layers to avoid confusion with neural networks, but they're kind of levels of organization, so to say. And what we see in brain tumor patients, but most people with a brain-related disease, is that you lose that organization. So the hubs become less connected. This is what also happens in terms of cognition. There's just less integration because these hubs become less connected. And at the same time, all these kind of leaf nodes on the bottom that would usually not be very connected, they are higher in local connectivity.
Do you find the same sort of pattern in other cognitive, like in non-cancer
for sure
cognitive problems?
This is the same for MS, Parkinson's disease, Alzheimer's, some psychiatric disorders.
Okay.
In general, this loss of integration is seen across, even in non-brain related diseases. Yeah. So people who have cognitive complaints or cognitive deficits typically also have lower integration of the brain network than sort of cognitively normal people.
Interesting. Like, what sort of, like, when I read what I got from Siddhartha Mukherjee's book, there's so many things, but one thing I was struck by is sort of that, like, for most cancers at least, it seems like what actually kills you is not necessarily the cancer itself, but the lack of the cells you need to do the job you needed to do. Like
the immune system.
Yes. So like.
Yeah, but I feel that's of course true.
Yeah.
But to some extent, it's also the problem is that that our bodies and our cells are so good at growing and maintaining function. That's how I see it in the brain. Because ultimately, what you see is that the cancer hijacks all these normal functions of brain cells. And that wasn't clear when I started at all. It's really been the past 10 years, but there's been major breakthroughs in the field, what's now called cancer neuroscience, such that there's this bidirectional relationship between the cancer cells and the cells around them. So both the supportive cells, the glial cells, obviously, because that's where the tumor originates from, but also the neurons. So there's actual synapses that connect the healthy neurons to the tumor cells, and each action potential, so each active brain cell makes the tumor grow faster.
What?
Yeah.
Wait.
Yeah.
Why would...
Because the cancer hijacks these normal functions. So in general, if a cell becomes active, a neuron, it says something because it got an input and it's being active, and maybe it wants to be plastic and connect to new cells or have stronger connections. This is the learning, adapting sort of skill of the brain that we maintain throughout life. But in this case, the cancer sort of picks up this normal activity.
But the cancer cell is not a neuronal cell, right?
It's a glial cell.
Okay.
But glial cells are also electrically innervated.
Oh, wow. Okay.
So basically, they transfer an electrical signal from a neuron into a calcium wave, which is the way that these oligodendrocytes communicate. Right. And this calcium wave is the way that the tumor cells then infiltrate and divide. Wow. Yeah. Yeah, so that's too bad. And it's bidirectional. So what we've known for a little bit longer is that tumor cells excrete glutamate, which is, of course, the major excitatory neurotransmitter, which makes the neurons around them more active. Okay. So we've seen that for a long time, that around a brain tumor, typically there's more like hyperactivity. And this is also one of the reasons why we see epilepsy in so many of the patients with brain tumors.
Okay, so I was thinking like... tumor cells are just like these dumb cells that can't...
they're very smart.
Oh, okay. They don't have any actual like skills to do something unique, but they actually have some stuff they can do.
Yes, for sure. And they hijack all of the functions, even from... Because these are adult patients. And they use sort of functionality that was more relevant in development.
Okay.
Like from when we were kids.
Right.
Because after that, these cells typically don't have this type of behavior. So they go back into
backwards in time.
Developmental stage, and then they can infiltrate more.
Yeah, so I was looking at this like review of just like this paper called Hallmarks of Cancer, trying to like sort of like lay out like few things that sort of all cancers sort of have in common. And one of the things that I read about is this sort of regression to doing things that you used to do when you were at a younger stage of development, because as cells grow, they specialize, they do their own thing, but they can also sort of go backwards and regress into this state where they're now sort of like they can do all kinds of other stuff that they're not supposed to be doing, and that sort of gives them the ability to go somewhere, take over and start doing some other different things.
Make new connections. That they are literally making new connections, probably.
Wow, that's crazy.
Yeah,
I like this is news to me. Okay, so let's say. Man, there's so much stuff. I didn't even get to ask. But I did want to at least ask about this grip-on fatigue thing. Yeah, I thought that was very interesting.
Yes, like we're very CBD, happy with that.
Okay, you need to say something about this, because for me, anytime like a psychological intervention has like this kind of effect is very interesting. Yeah, because this is like I wouldn't expect this.
No, many people didn't.
Okay.
Just to say let me sort of recap the whole thinking. So in order to get to grip on fatigue, I have to sort of go back into this localizationist idea, which I think is still very common, especially in neuro-oncology, because you have one apparent tumor, and most people think that everything that patients suffer from is due to the tumor itself, because it disrupts the tissue there, it breaks connections. There's of course lots of treatments, so most brain tumor patients get a surgery, large chunks of the brain are taken out, they get chemotherapy and radiotherapy, so there's lots of damage, that's what people think, and that can cause a lot of the sort of symptoms of the brain, of the brain tumor. So lots of patients have cognitive deficits, but something that is much more common even than cognitive deficits is fatigue. Okay. Like all other people with any sickness. Sort of fatigue is a general...
Like a physical... So, like a physical fatigue? Because I know
it's
there's
both.
Difference.
It's both. Okay. So we ask for 4 different aspects of fatigue in typical studies that we do in the brain tumor patients. It's not the best questionnaire for those fatigue experts out there, so we're aware. But we do ask for 4 different aspects. So one is mental fatigue, the other is physical fatigue, and a sort of concentration type attention fatigue, and then a general one. And you can make a sum score of that. And people are very fatigued, like over half of the patients have a severely fatigued
across the board.
Okay. But they are highly correlated. So there's almost no people who have like isolated mental fatigue, but no physical fatigue.
Because the other way around. Imagine it's hard to disentangle these two,
In general, I think the way we think about fatigue as like normal people typically is that you get fatigued because you sleep less than you're used to, or you exercise a lot, but the type of fatigue that you can have if you're not well. And I'm saying that in the most general sense. I mean, we do have it when we have a flu, for instance. You get so fatigued.
True.
And it's not clear to say where it's coming from, right? You're
exactly.
you're just like you
can't
move.
Like nothing is.
You can't think. You can't move.
Exactly.
So I think that's more like the fatigue that these people experience. It's kind of difficult to pinpoint where it is.
When I think of sort of like mental fatigue or like something, it's like I know I'm very sort of tuned to how I feel between a good night's sleep and a bad night's sleep.
Mm-hmm.
When I sleep anywhere less than like 7.5 hours, I feel like there's a part of my cortex I just can't access. It's like I just like some things like it's like people will say things to me and I'm just like it just goes in one ear and comes out the next. Like there's nothing happening.
And this has been proven. So sleep deficits make it very hard to be cognitively on par.
Yes.
There is the question whether you're actually so fatigued because your brain is not working or because you didn't sleep well.
Right.
And that sort of determines how you function during the day.
I know
because that's with the trackers.
They have a study on you tell people they slept better than they did and they just perform
They feel
better.
great.
They feel great.
Yeah.
Like, it's so ridiculous.
So there's a large, I don't think that psychology and the body are separate from each other. They are super connected to each other. So we can't see them as separate entities. And that's kind of what was something that we wanted to treat. So actually, I started this quest to treat this fatigue in these people because it was keeping them from working, from being a dad or a mom. It's really impairing their daily life. So of course they have a brain tumor. That's not great in general. But then if you ask them what's day to day, the most like crucial thing that is keeping you from living your best life, it's the fatigue and the cognitive deficits. So we wanted to treat that for a long time. And actually in, I think 2010 or something during my PhD, me and Martin Klein, who was my promoter, and I still work with him here in the hospital, we thought, let's just give them like stimulating medication. We gave them modafinil, which is kind of like methylphenidate, like Ritalin, to make them more active. And it totally didn't work.
But
so not only did it not work if you compared it to placebo on the tasks or on the questionnaires, everyone felt a little bit better because they got some pills. But the placebo did just as much better as the real deal.
Modafinil though is like I would have thought maybe like something like Ritalin or Adderall.
But it's very similar in how it works.
Okay. Dopaminergic sort of.
And since then people have tried also Ritalin or modafinil and many of these different things. It doesn't work. It doesn't treat the fatigue.
Wow, okay.
And people don't like it. So we had a very hard time getting people to participate because they don't want to take extra drugs in general.
I'm sure they're already taking enough for their...
They are taking anti-seizure medication usually. And they have a ticking time bomb in their brain. So actually any stimulants makes them feel like the buzz that you get from a stimulant. They don't like it because they're like, is the tumor growing? Is also something that's for these patients is hard. But I think in general, people have tried to treat like MS-related fatigue with these drugs. Doesn't work really.
Okay.
So, drugs didn't work. Then more recently, we came across Hans Knoop. He's a professor here also at Amsterdam UMC. He's a psychologist, and he developed this cognitive behavioral therapy for extreme fatigue. Okay. So I'm not sure what he started with, but once we started doing this in brain tumor patients, he had already shown that it works extremely well.
That's so crazy to
in
me.
All sorts of people, like people with CVS-ME, which is chronic fatigue syndrome, or like ME, what's it called? I forgot what the English word for the abbreviation is, but these are like severe fatigue syndromes.
Okay.
And people feel so much better. Like the effect sizes are
unheard of.
So when you say like severe fatigue, what are we talking about here?
I mean, like CFS is like a fatigue syndrome. These people cannot get out of bed.
Wow, that's crazy.
Yeah.
So okay, for all intents and purposes, they're like everything else is fine, generally speaking. Like,
I mean, there's lots of biological issues with these people. I'm not saying that it's. I'm not saying that they're fine, but they're not getting up or whatever.
Yeah.
But it's so. Let me broaden it some more, because they also looked at MS, multiple sclerosis, which is of course the degeneration of myelin in the brain, which makes muscles not like work well, and also cognitive deficits. Yeah. So this is a severe disease. People are very fatigued from that, but also palliative cancer. So people who have cancer, who are cannot be treated anymore and are in the final phases of life, they are also very fatigued. And in all these groups, this treatment helps. And the idea of cognitive behavioral therapy, for those who don't know what that is that it addresses the cognitions and the behaviors around a certain problem that you have. Yeah. And the idea with these diseases and fatigue is that there was somehow a trigger for severe fatigue. Like in our case, it will be the brain tumor. Yeah. A brain tumor is not helping your fatigue.
Yes.
Does not make you feel fit in general.
I imagine so.
Yeah, and especially the treatment afterwards, like chemotherapy obviously has a severe impact on your energy levels. So there's a clear physical trigger of these problems. The same goes for MS, for CVS. It's harder to say what it is, but we assume that there is a physical trigger that makes these people feel very tired. Once they feel very tired and they are diagnosed with a disease, I would add, they think, okay, so now I have a disease. And they have persisting thoughts and behaviors that keep this fatigue going. Even though the trigger itself, it doesn't have to be gone. I mean, brain tumors are never cured, so they're always there. But we think that the severe impact of the fatigue is persisting because of the cognitions and the behaviors. And these could be things like, there's nothing I can do about this fatigue because I have a brain tumor. So I'm just going to be fatigued for the rest of my life. The behavior could be poor sleep hygiene. So many people take naps during the day, which is useful if you're in the midst of chemotherapy. Well, once you're past that phase and you're kind of in the stable phase, it will be better not to sleep during the day, not to take naps. So there's this therapy has like 8 modules, I think, that looks at different aspects of the cognitions and the behaviors. Could also be, the original one that was developed by Hans Knoop did not have this one, I think, but like worrying about the future. Yeah. Because these patients with brain tumors they know it's going to come back usually. So they are worrying about the future, which can also be one of those cognitions that makes them tired. Right. And so we added that module, but the rest of them are, they sound super simple if you haven't seen the effect of CBT before.
The effect of CBT is crazy. Like I remember, like there was a study that showed that I think it's like just reading about CBT without actually doing it. Like just reading this book improves symptoms. Without actually
I haven't seen that one.
Without actually even doing it, I was like, okay, that seems ridiculous. I mean, that seems ridiculous, but like, it's I like it quite a bit. I try to do like because the cool thing is you can do CBT by yourself. Like you don't have to like
for sure.
You don't have to have a whole person to like.
And I mean, if you do think about it a little bit more, this is how we learn in life, right? Yeah. If you come into a problem in your relationships or when you're studying, you analyze what am I doing, and then you think, how can I do better? Yes. You're changing your cognitions and your behavior. Yes. So it's something we do all the time.
By the way, Claude and Gemini are surprisingly good at giving CBT advice.
Probably,
because surely they have just read so many books about it, right?
I mean, in general, it's not rocket science.
Yeah, it's not rocket science. So like, it helped me. Like it helped me quite a bit. Sometimes like my specific hang up is that like I tend to ruminate a lot, which is like classic something that's perfect to be addressed by CBT. And like it told me something super simple. It's like, okay, look, you've been ruminating on this for a week. If this line of thought was going to help you solve the problem, you would have found it by now.
Yeah.
So just stop. I was like, yeah, you're right.
Yeah.
I was like, wait, that's why didn't that occur to me before?
Yeah, and then there's the part, of course, that it's a reflex usually when you have, especially when you have these long-standing sort of tendencies like ruminating, probably will take you some time to also get rid of the reflex in addition to cognitively deciding, okay, it's not useful for me. And that's, I guess, so it's not rocket science, the things that people need to do, but the trick is to get them to do it because we have a pretty big resistance to accepting it is this simple.
Right.
To some extent,
people probably thinking that no, this is nonsense. Like,
yeah, and this is the whole key of the treatment, right? So as long as they keep believing that they will not get better because they have a tumor, it's not going to help because then they're blocking this effect of let's change my cognition. Yeah. So I'm not saying that we're solving all the issues related to fatigue because they still have a brain tumor.
No, it's like
but it. Like I can tell you the numbers, we included 40 patients and it was a randomized controlled trial. So half of them got the CBT. The other half was on the waiting list and they did get to do the treatment afterwards. So they did get the benefit just a little bit later than the ones who did get the randomized into the treatment arm. And of the non-treated patients, only 30% improved over the course of 14 weeks. That was the duration of the treatment. In the CBT arm, it was over 70%.
Well, that's big numbers.
Yeah, to put this into perspective, like even like cardial drugs for like blood pressure don't work as well.
That's crazy. Yeah,
so of course I'm saying it nicer than it is because this was an RCT. So we did select the patients to some extent that would benefit most. So I'm not saying that everyone will benefit from this and that it's completely comparable to blood pressure medication. But I'm just saying, like, as a first indication, this is huge.
Yeah, I understand. Like, I mean, the principle, if I'm understanding it correctly, is like, there's stuff about the tumor and the disease that you can't control. Yeah. It's out of your control. It will happen to you regardless. Yeah. But there's some stuff that you can control, which is like the thoughts and the behaviors that you have in response to that. There's like, I think what probably helps people the most in this situation is probably like this sense that there is some part of the progression of their life that is still under their control. Yeah. And it's not determined by this thing that just happened to happen to them. Like, you still there's still stuff that you can do to help yourself be better and feel better. Yeah. And I think that is just insanely valuable, right? For sure. And it does entail like people come, I think it's a blended CBT the way we did it. So that means that people came to the hospital for some face-to-face therapy sessions, I think 5 times. And then they also did a lot of homework on this online platform and got individualized feedback on the homework from the therapist. So it did take some work. Yeah. They had to do things every day. They had to practice, for instance, with their activity levels. So we would measure their activity levels before the treatment. And typically you would see 2 groups. And we did this with acti-watches and little mats in the when they sleep. But primarily the acti-watches. And then you can see that one group of patients, they are just very inactive in general, which makes you tired. We all know this. If you're healthy and you don't do anything for 4 days, you're going to be tired because that's the way the body works. Yeah, I remember like recently I sent my friends, I was just doodling. I sent my friends a graph of like, it's like a little stupid sketch. Like on the y-axis is the number of useful things you've done. And on the x-axis is like how tired you feel. Yeah. And it always starts like, I don't know, x-axis is how tired you feel at the end of the day. Mm-hmm. It's like no matter how relaxing of a day you have, you always have some baseline level of tiredness at the end of the day. So you might as well do something useful. Yeah, exactly. You know? So I was like, it's interesting. Yeah, so inactivity is not good for people in general. Yeah. But there's another group that is constantly over-pressuring themselves. So you would see a lot of activity and then they crash and then they have to lay in bed for 3 days and then they are doing way too much
and then they crash, and then they have to rest again. And both of these patterns are not useful. So the therapy would go into first determine which of the two someone is in, and then go to a like normalized, routine-based, like don't do too much.
It's quite adaptive. It's not like some blanket thing for
No,
everyone.
No, it's very individualized. So you do need a therapist who is able to do this.
Okay.
We had the measurements beforehand, so this is a quantitative measurement of their activity levels. So yeah, it is based on that for sure. And then you would sort of cut out the naps.
Would be crazy. Would be crazy if you just see. So I would imagine like this is effective. But I'm already seeing like a bottleneck is access to a psychologist and like an
actually Hans, so the PI who just championed this, is now working on getting it to be performed by specialized nurses, because there's just more of them.
I was thinking about AI because that's kind of what I'm working like.
I have severe doubts.
You think so?
Yeah, especially in our patient groups. I won't generalize to everything, but I think there has been studies on even like telehealth, which is online treatments, and it might work if people do it, but there's less inclination to do it. So the adherence is and compliance is just very low. Whereas if you have human contact and a better life for some people, but I think this can also be on the phone or in the video call, I think it makes a huge difference. Because people drop out of these purely, even if there is a human there, I think doing it only online, digitally, is not helping. We see that pretty clearly. So I think part of it can be sort of automatized to some extent, but having like human contact at set times, I think will definitely do a lot for this.
Do you think it's still like maybe it's not as effective, but it's still better than like imagine if the option was no treatment versus the same thing, but with like tele versus an AI doing doing the analysis and telling you what to do, what not to do. Like I would still think that'd be interesting to see, right? Like, or like if someone if it's like not 70% but like 50%, that's still better than.
I mean, at the group level, yes, it helps. It's better in a way. At the individual level, just way fewer people profit from it. So then the question is, of course, in terms of equity of care, if there are no CBT psychologists or registered nurses that have been trained to do this in your area or your country or even your continent, of course, then it's better to do it through an AI. But I think as someone working in healthcare, I would like to aim for everyone getting access, even if it's remote, to someone who can do this in person.
So the psychologist's role in the study was to tailor the type of type of like, so figure out what exactly the CBT they needed. Like okay.
So there was a general protocol, and the duration was the same for everyone, but the number and the specifics of the modules that would be followed would differ a little bit. Would be like between 5 and 8, I think. And the exact way in which it would be offered would be different.
Okay.
Yeah.
Okay, nice. That's
and you run
cool.
into, of course, lots of variations between people, because some people don't like online stuff. So I think for one person, we had to print out all of the assignments because they didn't like the online platform. So that could be a sort of a tweak that you do. Some people who have severe cognitive deficits, which is also likely in this patient group, would need more explanations than the average person participating. So these are all things that you can run into when doing this. And we're now looking at whether we can implement this at least all throughout the Netherlands. And there's also a center in Sweden looking whether they can implement the protocol.
Okay, so but I imagine this is generalizable outside of just. So wait, but has this already been shown for other conditions like
Yeah,
depression?
before we started.
Okay.
This,
I was thinking you could go like to all other things, but came the other way from other.
It came from other diseases like NMS, at least in the Netherlands. It's being used a lot. So there are trained psychologists in a lot of the different academic centers that perform this treatment for MS patients.
Okay, nice. Oh man, it's been an hour and a half.
Okay.
I
have a meeting.
Yes, exactly.
3.
Exactly. We'll wrap this up soon. But so you want to maybe say a few things about some things that you're particularly excited about in terms of research or some ideas in the future or some things you've seen in literature. Okay, maybe let's start with, is there something that you've learned that sort of surprised you or changed the way you think about something in the past 5 to 10 years maybe? I don't know. I don't know about this is a completely trial question. I've never asked someone this before. But I'm trying to get a metric for something where it's you've seen some unexpected sort of progress in much as in the field or something surprising. I mean, this is obviously really nice, the fatigue one, but anything in particular that you've seen in the past? I don't know, pick a time frame that you'd be comfortable.
Yeah, I think what we haven't talked about at all yet, but what is currently my most, the thing I'm most interested in and most inspired by, and also don't understand at all yet, is the way that issues tend to occur across different scales of measurement. So neuroscience typically is organized into different sort of silos. So there's people like me originally looking at behavior. Then there's people looking at scans, and there's people looking at cells or rats or, very model systems. And these are completely separate from each other. But we've been combining them over the past 5 to 10 years, all these different layers or sort of scales of measurement that you can look at. And we see the same things occurring from cells to networks to whole brains. So for instance, if you look at postmortem brains from donors with, for instance, MS, but also with Alzheimer's disease, you can see that people who have neurons that are larger and that have more dendritic density, so a more complex sort of cell, which hypothetically is more integrative at the very, very micro scale, they also have more integrated hubs in their brain network. And they are
size
also...
of the neuron?
Yeah, the size and the complexity of the dendrites.
Okay. Wow, that's crazy.
Yeah. And if we study this in patients who get surgeries, so that's one of the reasons why I think brain tumors are so interesting, but also epilepsy patients, because they get surgeries where they take out parts of the brain for therapeutic reasons. But then, of course, we can analyze these brain tissues. And then what we see the same thing. So patients who have more complex dendritic trees, so more density of the dendrites, more complex cells basically, also have more integration in the same region that the cells came from in the global brain network, which then correlates to their cognitive performance.
This is starting to me. The more I'm thinking about it, the more I'm trying to realize how sort of a deficient of a model the type of neural networks I work with are.
Well, it depends on what you want to model, right?
Yeah, exactly. I mean, when you teach someone, for example, neural network, when I was taught about it, you start out with there's always a compulsory slide with the neuron, with the biological neuron on it.
Yeah.
And I'm like, okay, now it's there's a size, there's a connectivity. I'm like, okay, well, maybe this is not the weighted sum, all right, but there's a bit more to this than...
For sure, and I would like to draw some attention to the supportive cells in the brain, because there is this neuron doctrine, right, that we think that everything, like the neurons are the basic units of the brain, which for some, like for electrical activity, they are, like for action potentials. But if you look at more complex processes, these glial cells are increasingly important. Microglia are really impacting lots of stuff. Yeah. And they do it in a less instantaneous way. So it's very difficult to measure. Yeah. But they impact lots of stuff. Yeah. So it's definitely not only the neurons that determine what is happening. Not for human, like general cognition, but especially in cancer. And that's, of course, what I'm looking at now. we're trying to understand how not just these cells interact with this bidirectional relationship between the neurons and the glial tumor cells, but also how that reverberates throughout the entire brain. So what we're seeing right now, and there's lots of people in the world doing like very high-tech cellular stuff, showing that these synapses, first we thought they were just glutamatergic. Now we know that they can also be GABAergic, serotonergic, dopaminergic. Probably all neurotransmitters are involved.
Okay.
So it's a very complex system where you have multiple types of connections between multiple types of cells. But what we do see in our studies where we combine measurements at these different levels is that the way in which the cells are organized, both the healthy cells and the tumor cells, relates to the way that the tumor functions within the global brain network and potentially also with the global network disturbances that we see in people, particularly with cognitive deficits. Okay. So we're kind of trying to tie that all together and see, of course, where we can treat.
Yeah, I mean, like...
Because that would be my ultimate game. I'm very fundamentally interested in the brain, but I've always been more also interested in helping people.
Yeah, of course.
So ultimately what I hope we can do is sort of perturb the brain at multiple scales in order to optimize what is happening.
Right. I mean, I think, like, for example, something simple, it seems simple, but the fatigue intervention.
Mm-hmm.
Like, I mean, first of all, your new center has a bunch of, like your lab is productive as hell. Like, I'm extremely, okay, I have to ask, the paper in a day.
Yeah.
How does that work? Like,
I have an awesome team, I have to say. So if you look at the newsletter, most of it is them, not me, if not all. So yeah, we've been practicing with the paper in a day. So basically what we do, and we've done this now 3 times for different papers, is that someone, usually a PhD candidate, who is at some phase of a project where there's results, and there has to be some, we typically pre-register our studies, so then you already have a layout of a paper or format. Generally, once that is there, we meet with a group of 2 to 4 people, and then we write the paper in one day.
What do you mean, write the paper? Right. But you need the results. You need data. You need how does that?
That's all there. So
okay.
It's not you start a project, then you write the paper.
Okay.
It's typically a year has passed.
Okay.
If you do data collection, years.
Yes.
And then once the point comes that you have the results, and
you just have to put the thing in the actual paper.
Yeah. So then you write the introduction and the discussion. I think that's more about conceptual frameworking, right? Write the methods and the results, which is more about what actually did I do and what is the order of that. Make figures and tables, and then sort of put it all together.
Yeah, I guess it's I no joke wrote my master's thesis in 2 weeks. Mm-hmm. 2 weeks meaning the actual writing, putting things in the report.
Yeah.
5 months of building code and everything. Yeah. But that's a super cool exercise. Why? Yeah. Have you noticed anything? What was the motivation for this? I like this idea. It's like a hackathon for a paper, right?
Yeah.
It's perfect. I love it. I think my friends would also love this. But
it's not my idea for sure. So I saw it somewhere else on back then Twitter.
Yeah.
But what attracted me to it is that I noticed that a lot of people have they work so hard at getting their experiments to work, collecting data in patients. That's hard, takes years. Then the moment comes that you do the analyses, and this is all going as you work towards something. But then once making the goal becomes closer, people sometimes slow down. And that's because it's hard to say, this is it, this is the paper, this is the end, because you can always keep going. And you can always say, did I do a perfect job? And no one ever does a perfect job. But that's, of course, especially if you're starting a PhD or if you're you haven't written that many papers yet, that's really a serious risk that you get stuck in perfecting, and not making the decision, okay, I'm just going to write it down now. I'll send it out for review and see what happens, And by doing this in a team-based manner, I think it makes it much easier.
Yeah. I can
So
see
it
that.
shortens this period of time where people are usually a bit lost. Like, is it finished? Is it not finished? Should I do some more analysis, or should I write it down?
They're saying the work expense to fill the time you have.
Yeah, for sure. And even if you don't have the time, because typically PhD candidates are well aware that the time is not endless, but it's just very hard to make that decision. And if you do it together, you can sort of feel like, okay, we went through all the options.
I can imagine this is super fun.
And it's super fun. It is super fun. And also because people can experiment with what they like to do. Yeah. Like, do they like making figures? Because then you can make figures all day. Yeah. Do you like writing? Then you can do writing. And of course, people circulate. We don't do the same thing always, but it's nice to try that and to be together and to focus on.
You guys do a lot of like fun stuff. Like I know, like the first few pages, I was like, oh my god,
yeah,
all over the place. It's awesome. Okay, so let's say to wrap this up real quick because I know you have a meeting also. Bottlenecks in terms of like, what are some like high leverage? Well, let's make it simple, like simple terms. Like, if you were like coming into this field, or like coming into, if you're a master's student, or let's say, master's student, right, and you want to try to meaningfully contribute something to do something, like it can be incremental, small little thing for experiments, but like, what is like a good place where 1 unit of time put might actually give us like quite a bit more in return.
Why would you frame it like that?
I have no idea. Like,
I'm
still like trialing. This is still like a trial in process. Like I'm trying to like.
You're trying to maximize results is basically your question.
No,
but I would turn it around.
Okay, go ahead. Tell me.
Yeah, so if you ask me like, what would your general advice be, or what is a bottleneck that I see in either young people or even in old people? I mean, where no one is excluded from this, but I think one of the things that is hampering this field that I'm in, like multiscale neuroscience about networks and interactions between oncology and neurology, is that people are stuck in their own frameworks. Okay. And this is not just thinking, because everyone has expertise that is local, right? Yep. So, of course, I talk now about cells, but that's because people told me how it works. I'm definitely not an expert on cells. So I'm much more comfortable if you ask me questions about cognition or brain scans. But regardless of that, in order to really understand how it works, I need to broaden my scope. Yeah. And it's scary because you're never an expert on everything. Yeah. But I think we need to feel the responsibility to do it if we want to progress in this field. Yeah. So, of course, we also need incremental steps within each respective fields. We still don't understand a lot about the cells. Yeah. We also still don't understand a lot about the fine, like nitty-gritty of cognition. So I'm not saying that there's anything wrong with working in those specific fields, but if you want to connect these different things, then you kind of need to throw away the boundaries between the fields.
As a concrete example, for example, you have for example, models of connectivity of networks in social science, let's say, and then you say, maybe I can frame the behavior in this other system. Yeah. For example, the brain, in the same way that they've characterized the behavior in this system, and then see if I can apply those techniques and models in that framework to this new
for instance. And that's why I'm using metaphors a lot, or like concepts instead of measures, like integration. That's of course confusing in the beginning, but it's a measure that I can apply to a network of the whole brain, because then I'm looking at path length. But I can also apply it to an individual cell, because then we know that a more complex dendritic tree is more integrative in its cellular network. So by using conceptual linkage, it makes it much easier for me to cross these different scales. Yeah. But it also hurts because you have to throw away some of the sort of specifics of each individual cell, because of course, or each individual level, because integration is not the same. It's not 100% the same thing. Integration between cells and integration between brain networks. I'm not saying it's the same thing. Yeah. But by using the conceptual similarity, I can draw parallels that we previously didn't draw. Yeah. and try to learn more.
Exactly. So are you familiar with Michael Levin?
No,
Like
I don't think so.
He's like crazy, incredible researcher in the US, and he does work on all kinds of, like his lab, they were growing limbs on frogs, and then the other PhD students are like looking at the emergence of intelligent behavior in simple algorithms, like simple sorting algorithms. So they showed like using models, why I brought this up is because they use models from a different domain, a different field, let's say cognitive psychology or whatever, or something like this, and they bring the model into this problem and then say, okay, can we characterize the behavior of this system using these techniques and see if we can learn something about it.
Yeah.
And then they show that simple algorithms like a sorting algorithm shows weird intelligent behavior. Like it'll reduce sortedness in the short term to increase sortedness in the long term. And you're like, wait, how? Because the algorithm itself has it's just a simple, if two numbers are similar, flip them around. Right?
Yeah.
But if you sort of mess with the system and say, well, sometimes the computer doesn't always switch the numbers, or like some numbers don't want to switch, then you find that all this weird behavior shows up, and you can characterize them with techniques from other disciplines.
Yeah.
So I understand. I think sort of what you're trying to say, maybe.
Yeah, and I think for that it's useful to have either a broad conceptual base, or have a shared methodological language. And for me, that's network theory, because you can apply it to everything, and because it mixes the different levels up. So that helps me a lot.
Okay, that's like that's a good bottleneck, I think. All right, we're gonna wrap this up. That was amazing.
Thanks.
Thank you so much for indulging me on a Friday. I'm sure you have better things to do, but thank you.
Not really.
No, not that.
Talking about science is kind of a good
awesome.
thing to do.
That thing is awesome, right?
Yeah.
But okay, thank you so much.
Thank you for having me.